Hello friends, today we are going to discuss one of the fundamental, important and very interesting topic from the paper of the genetics. Because all of us we know that in case of different types of organisms which are present on this earth, which includes different types of prokaryotic organisms, different eukaryotic organisms, there are archaea or archaebacteria also. So in this vast variety of microorganisms and macroorganisms which are present on this earth, there is a common phenomenon. So what is that common phenomenon which we observe in all these types of organisms or living beings which are present on this earth is gene regulation. So what is gene regulation? Because we know that in the genome of every living organism there are thousands of genes which are present over there, isn't it? And all those genes, they perform a particular activity which is necessary for the growth, survival, for multiplication of that particular living cell. So all the genes which are present in the genome, they should be controlled or regulated, isn't it? So that all the genes, they can perform a coordinated activity, and all such coordinated activities which are necessary for normal survival of the any living cell. Generally students ask about the multicellular organisms, say for examples in case of animals or in case of human beings, as one of the example of eukaryotic organism. We know that the questions asked by the students are like that: why sir only pancreatic cells they have been ability to produce insulin hormone? Why not any other body cells they do not produce insulin hormone, isn't it? We know that the insulin hormone is produced by the beta cells of islets of Langerhans, which are present in the pancreas. So why only pancreatic cells have ability to produce insulin hormone? Why not cardiac cells they produces insulin? Why not stomach cells or any other body tissues they have ability to produce insulin? Does it means that the insulin producing gene is not present over there? No, it is not like this. Insulin gene is present, or the gene which is responsible for production of insulin hormone is present in each and every living cell which is present in our body, isn't it? But even though that gene is present in the stomach cell, in the cardiac cell, in the brain cell, say for example, the gene is not going to be expressed over there. So that particular gene, why it is not expressing in other body cells, and why it is expressing only in the insulin producing pancreatic cells? So this is the general questions which are being asked by the many students. In the same case, in prokaryotic organisms as well, there are some genes which are active while some genes they remains inactive. So there are different types of mechanisms which actually control such a gene regulation. So our today's topic is to discuss about the regulation of gene expression. It is one of the important, fundamental and very interesting topic from any biological science point of view. The particular concept that we are going to discuss today is with respect to only prokaryotic organism. In eukaryotic organism, there are several mechanisms by which there will be gene regulation. In the same way, also in prokaryotic organisms, they also have a gene regulation. So under the title of regulation gene expression, we are going to discuss one of the subtitle which is specifically assigned with the prokaryotic system. And in that context, the title for my today's discussion or video is concept of operon. In that operon, we are going to discuss one of the fundamental and basic example of operon is the Lac Operon or lactose operon. So these are friends, these are my personal details. If you have any query regarding this particular video, you can directly call me or you can email me, or even you can put a comment on my YouTube video lecture. So we are going to discuss in this particular video lecture about the concept of operon under the subtitle of regulation of gene expression, isn't it? So first, gene regulation in prokaryotes, isn't it? In prokaryotic organisms, how genes are regulated? Because we know that all the genes which need not to be expressed for all the times. Even though there are thousands of genes present in the bacterial genome, all the genes they do not express at the time or simultaneously. Some of the genes they will be expressed, or some of the genes they will not be expressed. While some of the genes they have been constantly being expressed, which are important or have a vital function. If suppose the particular metabolic pathway is occurring in the bacterial cell, then if that metabolic pathway is very important from bacterial cell point of view, then the genes which are involved in the synthesis of enzymes which are required for that metabolic pathway, they are being constantly expressed, isn't it? While remaining genes, they express as per the need, We can see in the second point on this particular slide, all the genes, they are not expressed, while few genes are, they are expressed as per their need, isn't it? And this can be achieved. This can be means some genes they remain active, some genes they remain inactive. Means what? Some genes they are switched on, while some genes they are switched off. So how this particular achieved by the bacterial cell or prokaryotic system, and for this particular gene regulation there will be the concept of operon. So operon, it will explain why particular gene remain active or why it becomes inactive, and as per the need how gene will be regulated. And this concept of operon, it can be found only in the prokaryotic organisms. So what is operon? We will discuss in brief about the concept of operon first. So the word operon, we know that it has been coming from the Latin word operari. So what is operari? Operari means to work or to perform together. To work or to perform together in a coordinated manner. This is the meaning of operari, and from that operari word, the word operon is there, isn't it? Actually, the operari is terminology used in the Latin language for the musicians. We know that if there is a theatrical performance is there, in theater, if there is a musical concern is there, then all the musicians, they should play that music in a coordinated manner, isn't it? Then only that performance will be very beautiful, isn't it? And to perform all the musicians in a coordinated manner, there is a one person which we called it as a operari, and that operari, it helps in proper coordination in all those musicians, isn't it? And from that operari, or that from proper coordination terminology, the term operon was given there. It was come from the Latin word. So what is operon? By definition, we can say that operon is an operating unit. What is operon? Operon is a operating unit which can be defined as the cluster of genes which are located together on a particular chromosome. Operari means what? These are the cluster of genes which are present on the particular chromosome, and they transcribe together to perform a particular activity. They transcribe together. Together means what? All the genes which are coming under a single operon, they transcribe together so to perform a coordinated activity over there. They usually control an important biochemical process. The role of operon is to control the important biochemical process. The concept of operon, it was first described in 1961 by Francis Jacob and Jacques Monod of the Pasteur Institute of Paris. And for this work, Jacob and Monod, they won the Nobel Prize in Physiology or Medicine in 1965, isn't it? So first we will discuss in brief about the general structure of operon, why it is called as a cluster of genes, or how the concept of operon is helpful in understanding how the gene has been regulated in the unicellular organisms, or in case of generally prokaryotic organisms. So if we consider the general structure of operon, we can find there are different types of genes in that particular operon. The first gene that we are going to discuss is the repressor gene. The name itself indicates that the repressor gene is responsible for producing a repressor protein. And what is the function of that repressor protein is to fit into the operator to turn operon off, isn't it? The repressor gene will produce a repressor protein. That repressor protein then will bind to the operator region, and that operator region, once it get bind with the repressor protein, then the operon, it becomes switched off, or the gene coming under that particular operator region will remain inactive. So this is the repressor gene. Second one is the promoter. It is another important part of the general structure of operon. Promoter, what is promoter, or what is its role? The RNA polymerase attaches to the promoter. And once RNA polymerase attached to the promoter, then promoter will help in the transcription of downstream genes. Isn't it? Downstream genes means those genes which are present on the right side of the promoter, isn't it? And promoter, what is the promoter? Function of promoter? Promoter provides binding site for the RNA polymerase, and we know that RNA polymerase is essential for transcription of genes, isn't it? Because once the genes have been transcribed, they will be further translated to produce desired protein, and that is the function of promoter. The next one is the operator. As I said, in the repressor gene, operator, it provides binding site for the active repressor molecule, and once the repressor molecule will bind to the operator, remember one thing, that once the repressor it will bind to the operator, the RNA polymerase cannot bind to the promoter, and therefore when the repressor protein bind to the operator, the genes, they are not going to be transcribed because RNA polymerase is not allowed to bind to the promoter when the operator is bound with the repressor molecule. And the final part, last part, is the structural genes. These are the actual metabolic pathway genes which codes for different types of enzyme proteins which helps in the digestion of lactose. I've been described it here specifically because in next point we are going to discuss the particular operon, that is Lac Operon or lactose. So what is the role of structural genes? Structural genes, when they transcribe, they produces particular types of enzymes which are necessary for the digestion of particular type of metabolite. So we can see here the general structure of operon. As I said, we can see here the regulatory gene, isn't it? What is the function of regulatory gene? Is to produce repressor protein. And what is the function of repressor protein? We can see here the repressor protein, it can bind to the operator. The blinking arrow indicating that the repressor protein, it can bind to the operator. And once the repressor protein, it will bind to the operator, as I said previously, RNA polymerase cannot bind to the promoter. And there will be no transcription and the operon, it remains switched off. You have to remember this, isn't it? The next part is the promoter. After regulatory gene, it is the promoter. What is the function of promoter? Promoter, it allows the binding for the RNA polymerase enzyme. But when RNA polymerase can bind to the promoter, when there will not be a repressor protein binding to the operator. Means what? Once operator site remains free, then and then only RNA polymerase, it can bind to the promoter. The next last part is the structural genes. You can see there highlighted arrow indicating the structural genes. Say for example, here 4 structural genes, they have been shown in this particular general structure of operon. These are A, B, C and D. These are the structural genes. What is the function of structural genes? When RNA polymerase binds to the promoter, then that RNA polymerase will transcribe those structural genes present on the DNA or in that particular operon region. And all the ABCD genes, they undergoes the process of transcription with the help of RNA polymerase to form mRNA, and mRNA will be finally translated to produce 4 different types of proteins like protein A, B, C and D. And all those different types of proteins, they are involving in the particular activity. So whenever there is a need, then and then only those proteins or enzymes, they will be synthesized by the bacteria. Because we know that each and every living being cell is very much cautious about their economics. What is cell economics? Economics is nothing but each and every living cell, it try to conserve their ATPs. So until or unless the enzymes are necessary, then and then only the operon will remain active. If it is not needed, then operon, it remains switched off. It remains inactive or switched off if there will not be need of those particular enzymes. Isn't it? And that is the kind of gene regulation which is occurred with the help of concept of operon. And this is the general structure of operon. We can see a regulatory gene, promoter, operator and structural genes. Isn't it? Now we are going to discuss a specific operon as an example of gene regulation, that is lactose operon. So what is lactose operon? It is generally being designated as a Lac Operon. Lactose operon is designated as a Lac Operon. The Lac Operon is responsible for coding different types of enzymes which are actually involved in the catabolism of degradation of lactose. Isn't it? And what is lactose? Lactose, we know that it is a disaccharide. Lactose is a disaccharide which is made up of by glucose and galactose. And the example of lactose operon is an example of inducible operon. It is inducible operon. So why it is called as an inducible operon? Because the presence of lactose, it will induces the operon to remain active. Isn't it? It is called as inducible operon because the presence of lactose induces that operon to become active or to become switched on. Means what? In presence of lactose only, the Lac Operon will remain active. In absence of lactose, obviously it will remain inactive or switched off. And therefore it can be called as a inducible operon. You can see the general structure of Lac Operon. You can see their repressor gene or regulatory gene, isn't it? It can be labeled as a regulatory gene I or repressor gene I. Then regulatory region, it also includes 2 another genes, operator and promoter. You can see there. And right side to the operator region, there are 3 structural genes. The name of those 3 structural genes is the LAC-Z, LAC-Y and LAC-A. And these 3 structural genes, they are responsible for the production of 3 different types of enzymes. isn't it? So this is the general structure of Lac Operon or lactose operon. And as I said previously, lactose operon is responsible for synthesis of those enzymes who are involved in the metabolism or degradation of lactose, and that operon remains switched on only if the lactose is present, as it is an example of inducible operon. So what are those three genes or structural genes, and what is their enzyme? So first structural gene is the LAC-Z gene, which is responsible for coding an enzyme beta galactosidase. And what is the role of beta galactosidase? Is to break down the lactose into glucose and galactose. The second structural gene is the LAC-Y gene enzyme, which is responsible for coding enzyme permease. As the name itself indicates, this particular permease enzyme is located in the cytoplasmic membrane of the Escherichia coli cell, and that membrane protein, it allows the permission for the lactose to come inside the cytoplasm from outside of the medium. And third one is the LAC-A gene, which is responsible for production of transacetylase enzyme. The function of this enzyme is to transfer, the name itself indicates, this enzyme transfers acetyl group from acetyl-CoA to the galactose side. So all these are the three important structural genes which are responsible for coding three different enzymes, and all these enzymes together involved in the lactose metabolism. And when these three genes are going to be expressed? In presence of lactose only. Otherwise, these three genes will not be transcribed or will not be expressed. That case, we can call it as a switched off case of that particular operon. So how regulation of operon will take place? We are going to discuss in this particular point. Regulation of Lac Operon, if we consider the first situation when lactose is present or lactose is absent. In the first situation, we are going to consider about when lactose is absent. And lactose here, it works as like that of inducer, because the presence of lactose induces operon to remain active or to switch on. So when lactose or inducer is absent, from cell point of view, we are going to think, then obviously cell will not produce enzymes which are involved in the lactose metabolism, because there is absence of lactose. So in absence of lactose, initially we should know that the operon, it remains switched off. So what is the detailed mechanism or how that operon becomes switched off in absence of lactose? We are going to discuss now. So in absence of lactose, what will happen? We can see here, when lactose is not there, the regulatory gene is responsible for production of repressor protein. So this is the situation. When lactose is not there in the surrounding medium, then the function of regulatory gene is to produce repressor protein. And what is the role of repressor protein? That repressor protein, it binds to the operator. What is the function of repressor protein? Repressor protein, it will bind to the operator site and prevents the binding of RNA polymerase to the promoter site. And if RNA polymerase is not being able to bind to the promoter site, obviously structural genes will not be transcribed. You can see in this particular diagram, isn't it? And if the structural genes are not transcribed, then what will happen? What will happen? The gene, if in the absence of transcription, there will not be activation of expression of structural genes, and the mRNA will not be produced, and those enzymes, they are not produced in absence of lactose, isn't it? In another situation, if suppose lactose is present, have you understood? Or you can see repeatedly it. See in this particular slide, you can see in this particular image, you can see in the absence of lactose, the repressor gene will be expressed to produce repressor protein, and you can see there that repressor protein, it can bind to the operator region, isn't it? And that operator region, once bound with the repressor protein, there will not be binding of RNA polymerase to the promoter, and therefore structural genes of Lac Operon, they will not be transcribed, and they will not be produces any enzymes which are required for the lactose metabolism, because there will not be presence of lactose in the surrounding medium. So this is the case in absence of lactose. So what will happen if the lactose or inducer is present, isn't it? When lactose is present, if we think from the cell point of view, then obviously cell now start to produce different types of enzymes which are involved in the lactose metabolism, isn't it? So when lactose or inducer is present in the medium, then obviously the lactose or inducer, it will bind to the repressor, because the repressor is whole and solely responsible for the operon to remain switched off. But when the lactose inducer is present, it will bind to the repressor molecule, and what will then happen? Then repressor, it fails to bind to the operator. You can see in this particular diagram, isn't it? There is an inducer, lactose, that red colored dot shown over here, that inducer lactose is bind to the repressor, and the complex of repressor inducer, it doesn't bind to the operator, isn't it? It doesn't bind to the operator. You can see in this particular diagram that inducer repressor molecule, which is a complex formed over there, and that complex, it doesn't bind to the operator region, isn't it? And once the repressor is not being able to bind to the operator, what will happen? Then operator remains free, and once the operator remains free, now RNA polymerase being able to bind to the promoter. And once the RNA polymerase bind to the promoter, you can see in this particular diagram, there will be the transcription of LAC-Z, LAC-Y, LAC-A gene. And once the transcription takes place, then three different types of enzymes, they will be produced, like beta galactosidase. The function of this particular enzyme is to degrade the lactose into glucose and galactose. Then it will produce as permease. Permease, it allows the entry of lactose inside the cell. Then only beta galactosidase can act, and the transacetylase, which will transfer the acetyl group to the galactosides, isn't it? So the mRNA, which is transcribed after transcription, which is produced, which further codes for these three particular enzymes which are required for the lactose metabolism. So this is the case when the lactose inducer is present. In another situation, what will happen? We can consider this another situation, that when both glucose and lactose are present. So what will happen in presence of both glucose and lactose sugars? If suppose E. coli is present in the nutrient broth medium, and we have supplied both glucose and lactose sugars to the, so do the cells of E. coli use both glucose and lactose simultaneously, or one at a time? This is the question. If E. coli cell is allowed to consume both glucose and lactose, what E. coli cell can do? Whether E. coli cell utilizes both the sugar simultaneously or one by one? The answer to that particular question is that the Escherichia coli cell only utilizes glucose first, not utilizing lactose if glucose is present in the surrounding medium. The answer is that glucose is utilized first. So why glucose is utilized first? Because it would be wasteful to induce the enzymes which are required for the sugar other than the glucose, because glucose can be readily assimilable. It is very important, or it can be readily utilizable as a source of carbon and energy. Cell need not to produce any additional enzyme to utilize the glucose. Glucose can be directly assimilated, isn't it? If cell can utilizes glucose very easily, then why Escherichia coli cell it can utilize lactose then? So E. coli cell will prefer the glucose in presence of lactose, isn't it? Because E. coli cell can grow faster on the glucose. Because we know that for the utilization of lactose, E. coli first have to produce enzymes required for the degradation of lactose, then only E. coli cell can utilize lactose. This is not the case for glucose, isn't it? And therefore glucose first will be preferred as a source of carbon and energy by the Escherichia coli in presence of lactose. And this particular phenomenon is called as a catabolite repression. We can call it as a catabolite repression. It is a mechanism of control or global control mechanism that decides between different available carbon sources. If more than one carbon sources are present, then the catabolite repression decides which carbon source is to be first utilized. In this particular catabolite repression mechanism, what is catabolite? Here glucose is catabolized. And why it is called as a catabolite repression? Because in presence of glucose, the enzymes required for the lactose utilization, they will not be synthesized. So in presence of glucose, lactose operon can be repressed, and therefore this can be called as a catabolite repression, isn't it? That catabolite repression sometimes also called as a glucose effect. So why it is called as a glucose effect? Because glucose was the first substance shown to cause this particular response. Therefore it is also called as a glucose effect, and thus catabolite repression ensures that organism can only utilize easily available carbon and energy source first, and they, after the exhaustion of glucose, then organism they can go to utilize for another sugar which is present here is lactose, isn't it? And this is achieved with the help of catabolite repression. This is the case actually when both the sugars are present, isn't it? In the last point, we are going to discuss in brief about Lac mutations. What is Lac mutations, isn't it? If there are alterations takes place in the Lac Operon, they can be called as Lac mutations. Say for example, if there are alterations or mutations in the Operari region, because Jacobian monad find some constitutive mutants. Why it is called as constitutive? These constitutive mutants have to be mutated at their operator site, and such a Lac mutants which have a mutations in the operator site, they have been called as a Lac OC. C for constitutive isn't it? And mutations takes place in the operator region, therefore they are called as the lac OC. And why they are constitutive mutants they are being called? Because we know that if there is mutations in taking place in the operator region, then repressor molecule cannot bind to the operator, such altered operator, isn't it? And once the repressor is not being able to bind to the operator, then promoter region constantly being free for binding to the RNA polymerase. And if RNA polymerase is constantly bind to the promoter, then what will happen? Then the structural genes, they being constantly expressed, isn't it? The structural genes, they become constantly expressed, and therefore it is called as a constitutive mutant, because such a mutants with mutations in the operator regions, they found to be constitutively synthesize beta galactosidase enzyme, even though lactose is present or absent, isn't it? Then second lac mutations are the promoter mutations. The name itself indicates if there will be mutation in the promoter, what will happen? If there is a mutation in the promoter, then obviously RNA polymerase enzyme, it cannot bind to the promoter region, isn't it? And such a mutations, if they are taking place in the promoter region, then such a mutants, they are being called as a lac P minus. What they are called as a, these mutants are a designated as lac P minus, isn't it? Because they have a mutations in the promoter regions, and therefore altered promoter region will not bind to the RNA polymerase enzyme. And what will happen then? Because we know that the binding of RNA polymerase to the promoter is essential for the expression of structural genes. If there will be the mutation in the promoter, then obviously RNA polymerase, it cannot bind to the promoter, and therefore E. coli cells with such a lac P minus mutations, that does not produce enzymes, because RNA polymerase not been able to bind to the promoter. And if enzymes, they are not produced even in presence of lactose, they can be considered as a lac mutations in the promoter regions, because even though lactose is present, the cell will not be producing lactose utilizing enzyme, isn't it? And such mutant, they are called as a lac P mutant. So this is in brief about the concept of lactose operon, and in addition we have discussed in brief about lac mutations. So these two kinds of mutations, they have been commonly observed, isn't it? So thank you for watching this particular video. If you like video, do share, like and subscribe, isn't it? Thank you very much.